Hello there, and welcome to the Dreamy Ocean Channel. As the day's light begins to soften, allow your breath to deepen a slow, gentle rhythm that eases through your entire being. Feel your shoulders relax, releasing any lingering tension, letting your jaw gently unclench. Allow the subtle weight of your body to settle, surrendering to the quiet comfort of this moment, as if you are gently floating, suspended and calm. Tonight, we journey into an extraordinary realm far beneath the familiar waves where sunlight cannot reach. Beneath the tranquil surface of our vast oceans, a realm of profound mystery awaits, holding secrets of life. So extraordinary, some have yet to be officially whispered into our textbooks, lingering instead in the hushed archives of scientific exploration. Imagine creatures glimpsed but never fully described, specimens held in silent contemplation, their existence known yet unannounced to the wider world. These are the untold stories of the deep, the hidden wonders that beckon us into a realm where silence reigns and discovery continues perpetually just beyond our full grasp. Let your mind now gently descend with us into these enigmatic, dreaming waters. The unseen ocean's vast embrace. The gentle descent begins a slow, deliberate passage away from the sun-drenched surface waters, where the familiar hues of sapphire and emerald give way to an ever-deepening indigo. With each meter that passes beneath us, the vibrant energy of the upper ocean, teeming with photosynthetic life, gradually recedes, leaving behind a profound tranquility. The warming embrace of the sun's light, which fuels the planet's surface ecosystems, diminishes with a quiet surrender, its spectrum absorbed by the vast liquid above. First, the reds and oranges are swallowed by the immense volume of water, then the yellows, until only the cool, ethereal blues and violets persist, painting the world in monochromatic shades of twilight. This is not a sudden plunge into darkness, but a graceful, elongated fade, a gradual transition into a realm where the very concept of light as we know it ceases to hold sway. As we continue our imaginary journey downward past the sunlit, epipelagic zone where most of marine life thrives, we enter the mesopelagic or twilight zone, a vast expanse stretching from approximately 200 meters down to 1,000 meters. Here, the last vestiges of sunlight, filtered and weak, penetrate like distant memories, barely illuminating the water column. It is a world of perpetual dusk, where the forms of creatures begin to blur and shadows stretch long and indistinct. The pressure, an unseen but ever-present force, begins its subtle assertion, increasing steadily with every 10 meters of depth. For every 10 meters we descend, the pressure intensifies by 1 atmosphere, a silent, pervasive weight that compresses and molds the very fabric of this watery universe. On the surface, our bodies exist under the weight of a single atmosphere, but here in the mesopelagic, it can reach 100 atmospheres, a force that would crush most terrestrial life instantly. Beyond the mesopelagic, the journey continues into the bathypelagic zone, a truly abyssal expanse commencing at 1,000 meters and extending down to 4,000 meters. This is the realm of absolute darkness where the sun's light cannot penetrate at all. Here, the world is lit only by the sporadic, often spectral glow of bioluminescence, an internal light produced by the creatures themselves, shimmering like isolated living stars in the profound blackness. The temperature also stabilizes dramatically in these depths, dropping to a near-freezing uniform chill, typically between 0 and 4 degrees Celsius. This cold is constant, unchanging, a testament to the ocean's vast thermal inertia, a deep, pervasive coolness that permeates every molecule of water. The pressure, too, continues its relentless increase, reaching up to 400 atmospheres, equivalent to stacking 40 jumbo jets onto a single human thumb. Yet life persists, exquisitely adapted to this crushing environment, its very structure altered to withstand forces that would shatter lesser forms. Further still below 4,000 meters lies the abyssal pelagic zone encompassing the vast flat plains of the abyssal seafloor. This immense, largely unexplored region covers more than half of the Earth's surface, a colossal, featureless plain of silty sediment stretching for thousands of kilometers. Here, the pressure can exceed 600 atmospheres, a profound, unimaginable weight that presses down from every direction. The environment is characterized by extreme cold, perpetual darkness, and an almost complete absence of nutrients raining down from the sunlit surface. Food in these depths is a precious commodity arriving primarily as marine snow, a slow, continuous drift of detritus dead organisms, and fecal pellets from the upper layers, a gentle, ethereal snowfall that sustains the sparse yet uniquely adapted communities below. This delicate shower of organic matter is the primary energy source for most abyssal life, a lifeline from a world far above. And then, in the deepest trenches, the ocean plunges even further into the hadal-pelagic zone, a realm of extreme depths found within the ocean's deepest canyons, and trenches like the Mariana Trench. These narrow V-shaped chasms, often exceeding 6,000 meters in depth, represent the most extreme environments on Earth, reaching down to nearly 11,000 meters. Here, the pressure can exceed 1,000 atmospheres, a force so immense that it is difficult for us to truly conceptualize. Imagine the weight of an entire elephant balanced on the tip of your finger, intensified 1,000-fold. This is the kind of pressure that organisms in the hadal zone endure, their cellular structures and biological processes finely tuned to resist such colossal forces. The water, though dense and cold, is relatively uniform, lacking the dramatic currents and turbulence of the upper ocean, creating a world of profound stillness and quiet. The sheer scale of this unseen ocean is difficult to grasp. While humanity has extensively mapped the surface of the Moon, and Mars, less than 1/5 of our own ocean floor, has been accurately charted with modern sonar technology, vast expanses remain entirely unknown, their topography and inhabitants speculative. This immense uncharted territory represents the planet's largest contiguous habitat, a colossal volume of water and sediment stretching across the globe, holding secrets that have remained undisturbed for eons. The average depth of the ocean is approximately 3,700 meters, meaning that the majority of our planet's liquid realm exists in perpetual darkness under immense pressure and at near freezing temperatures. This deep ocean is a world unto itself, disconnected from the rhythm of day and night seasons and surface weather patterns, a stable, ancient environment where evolution has unfolded along paths entirely alien to our terrestrial understanding. The profound silence of the deep ocean is another defining characteristic, a pervasive quietude broken only by the whispers of geological activity, the distant rumble of submarine earthquakes, or the faint, almost imperceptible clicks and groans of deep-sea creatures communicating in the darkness. Sound travels differently through water, much faster and further than in air, yet the vastness and the absence of constant surface noise create an overwhelming sense of isolation. Here, the absence of light means the absence of visual cues, making sound and chemoreception, the ability to detect chemical signals, the primary senses for navigation, hunting, and communication. Organisms have evolved extraordinary adaptations to perceive their world in this sound-rich, light-poor environment, developing highly sensitive lateral lines, complex bioluminescent patterns, and intricate chemical sensing organs, all designed to navigate and survive in a realm where vision is a luxury, not a necessity. The sparse distribution of food in the abyssal and hadal zones has driven the evolution of unique feeding strategies. Many creatures are ambush predators, conserving energy and waiting patiently for the rare passage of marine snow or unsuspecting prey. Others are scavengers drawn to the occasional colossal windfall of a whale fall, a nutrient-rich feast that can sustain an entire community for years. Still others have developed symbiotic relationships with chemosynthetic bacteria, particularly around hydrothermal vents and cold seeps, where chemical energy rather than solar energy forms the base of the food web. These oases of life, fueled by geothermal activity and chemicals seeping from the Earth's crust, are stark reminders that life finds a way even in the most extreme conditions, completely independent of the sun's distant warmth. They are vibrant, unique ecosystems teeming with species found nowhere else on Earth, a testament to the adaptability and resilience of life. This environment, so alien and yet so fundamental to our planet, has only recently begun to yield its secrets to human exploration. For millennia, the deep ocean remained an impenetrable mystery, its depths guarded by insurmountable pressure and darkness. Early attempts at exploration were fraught with peril, limited by technology and understanding. It was not until the invention of robust submersibles and remotely operated vehicles capable of withstanding the crushing pressures and navigating the perpetual night that humanity could truly begin to glimpse the wonders hidden beneath the waves. Even with these technological marvels, the sheer scale of the deep ocean means that only a minuscule fraction has been directly observed or sampled. Each dive, each remotely operated vehicle deployment reveals new species, new behaviors, and new insights into the fundamental processes of life on Earth. Yet for every creature documented, countless others surely remain undiscovered, unseen, their existence known only to the silent currents of the deep. This vast, unexplored frontier stretching across the majority of our world is a powerful reminder of the limits of our current knowledge. It is within these colossal, silent depths, under unimaginable pressures and in perpetual darkness, that some of the most extraordinary and enigmatic forms of life are believed to reside. Creatures that defy conventional understanding, their forms and functions honed by eons of isolation and extreme conditions. It is here, in this profound abyss, that the boundaries of known biology are continuously challenged, where specimens are sometimes glimpsed, captured, and then for various reasons remain unannounced, quietly held in the archives of scientific expeditions, awaiting their moment of formal recognition, or perhaps forever to remain a whispered secret of the deep. This immense, unseen ocean, therefore, is not merely a physical space, but a repository of biological mysteries, a dreaming ocean where the unknown vastly outweighs the known, inviting us to ponder the wonders that still lie beyond our grasp, waiting for their stories to be told. Navigating the deep's hostile environment, the colossal, unseen weight of the ocean presses down, a pervasive force that shapes every aspect of this submerged world, defining its very existence. As we drift deeper beyond the farthest reach of the sun's gentle touch, the hydrostatic pressure becomes the omnipresent architect of life and landscape. It is not a force that pushes from a single direction, but rather an encompassing embrace, pressing equally on every surface, every molecule, every cell from every conceivable angle. Imagine the subtle yet relentless tightening of an invisible hand, slowly, inexorably compressing everything within its grasp. At the average depth of the ocean, approximately 3,700 meters, The pressure exerted by the overlying water column is roughly equivalent to 370 atmospheres. This means that every square centimeter of surface area is subjected to a force of about 370 kilograms, a staggering burden that would instantly flatten most forms of life accustomed to our surface world. For human explorers and their intricate machines, this crushing pressure presents the most formidable barrier to discovery. Our familiar air-filled spaces, lungs, sinuses, even the minute air pockets within our cells would be immediately collapsed and destroyed. The gases within our bodies would compress dramatically, leading to excruciating pain and rapid loss of function. Even the most robust conventional materials like steel or aluminum would deform and buckle under such immense stress, their molecular bonds straining against the overwhelming external force. To penetrate this realm, engineers must construct vessels of extraordinary strength and resilience, crafting spherical submersibles from materials such as ultra-high-strength steel alloys, titanium, or advanced ceramics. These meticulously designed habitats are engineered to evenly distribute the colossal pressure, transforming a deadly external force into a manageable uniform stress across their curved surfaces, allowing a fragile bubble of atmospheric pressure to exist within the abyss. Each weld, each joint, each viewport is a triumph of material science, a testament to humanity's yearning to glimpse the unseen. Yet, for the indigenous inhabitants of the deep, this pressure is not a hostile anomaly, but the very condition of their being. They are not merely surviving it, they are perfectly integrated with it. Many deep-sea creatures, particularly invertebrates, and certain fish, lack gas-filled organs like the swim bladders that allow shallower fish to control buoyancy. Instead, their bodies are largely composed of water and incompressible tissues, allowing them to exist in perfect hydrostatic equilibrium with their surroundings. Their internal pressure matches the external pressure, eliminating the destructive differential. Their tissues are often soft, gelatinous, and highly flexible, yielding to the pressure rather than resisting it, allowing them to move with an elegant, almost ethereal grace through the dense liquid. Imagine a creature whose very substance is designed to flow with the unyielding current of pressure, rather than to stand against it. At a cellular level, the adaptations are even more profound and subtle. The proteins within the cells of deep-sea organisms are specifically evolved to function optimally under extreme pressure. On the surface, high pressure can denature proteins, causing them to unfold and lose their biological activity, much like excessive heat. However, in piezophilic or pressure-loving organisms, these proteins have evolved unique structures and amino acid sequences that ensure their stability and proper function, even under hundreds of atmospheres of pressure. Furthermore, many deep-sea species accumulate high concentrations of osmolytes within their cells, particularly trimethylamine N-oxide or TMAO and other compatible solutes. These organic compounds act as molecular chaperones, stabilizing proteins and counteracting the deleterious effects of pressure on cellular machinery. They are like tiny internal shields, protecting the delicate biological processes from the crushing forces that surround them. The presence of these osmolytes allows the enzymes that drive metabolic reactions to continue their work with serene efficiency, despite the profound physical stress. Beyond the relentless pressure, the deep ocean presents a world of perpetual, absolute darkness, a profound absence of light that sculpts sensory evolution in extraordinary ways. Beyond the faint blue-green glow of the mesopelagic, the bathypelagic and abyssal zones are truly aphotic, devoid of any light originating from the sun. Here, the only illumination comes from within, generated by the organisms themselves through the mesmerizing phenomenon of bioluminescence. This internal light produced by chemical reactions within specialized organs called photophores, or by symbiotic bacteria, serves a multitude of purposes. It can be a lure, a shimmering beacon drawing unsuspecting prey closer to awaiting more, as seen in the infamous anglerfish with its glowing esker. It can be a defensive flash, a sudden burst of light designed to startle a predator, or to create a burglar alarm effect, attracting a larger predator to consume the attacker. Or it can be a subtle coded signal, a silent language of light for communication courtship and species recognition in a world where visual cues are otherwise non-existent. Some deep-sea creatures, particularly those residing in the upper reaches of the bathypelagic, where the last faint echoes of surface light might still linger, possess extraordinarily large tubular eyes meticulously adapted to capture every stray photon. These eyes often point upwards, scanning for the faint silhouettes of prey against the even fainter downward filtering light, or for the telltale flashes of bioluminescence. Yet deeper still, where light is truly absent, eyes may become vestigial, small, and non-functional, or disappear entirely. Energy is a precious commodity, and maintaining complex visual organs in a world without light would be a wasteful expenditure. Instead, these creatures rely on other senses finely tuned to the nuances of their environment. Chemoreception, an acute sense of smell or taste through water, becomes paramount, allowing them to detect the faint chemical trails of prey or potential mates across vast distances. Mechanoreception, the ability to sense vibrations and pressure changes in the water through lateral lines and sensitive tentacles, aids in navigation and predator detection in the profound darkness. The pervasive cold is another defining characteristic of the deep ocean, a constant near-freezing chill that further slows the pace of life. Below the thermocline, where surface waters meet the deep, stable layers, temperatures typically hover between 0 and 4 degrees Celsius. This extreme cold significantly reduces metabolic rates, leading to slower growth, longer lifespans, and reduced energy requirements for many deep-sea organisms. Their biological processes operate with a serene, almost languid efficiency, conserving precious energy in an environment where food is exceptionally scarce. Unlike surface dwellers, deep-sea fish do not experience dramatic temperature fluctuations, allowing their enzymes and cellular machinery to be perfectly optimized for this narrow, frigid range. While not as universally present as in polar fish, some deep-sea species do possess antifreeze compounds in their blood and tissues, preventing the formation of ice crystals at temperatures that could otherwise be lethal. Indeed, the scarcity of food is a fundamental challenge shaping deep-sea ecosystems. Far from the sunlit surface where photosynthesis fuels the food web, the deep ocean relies almost entirely on the slow, continuous descent of marine snow, a gentle, ethereal shower of organic detritus from above. This sparse, unpredictable rain of dead plankton, fecal pellets, and decaying organisms provides the primary energy source for the vast majority of deep-sea life. Consequently, creatures have evolved remarkable adaptations for capturing and utilizing every scrap of available energy. Many are ambush predators equipped with enormous expandable jaws and stomachs capable of swallowing prey much larger than themselves in a single, infrequent meal. Others are patient scavengers drawn to the rare, colossal windfall of a whale. Fall, a nutrient-rich feast that can sustain entire communities for decades. The slow, deliberate movements and extremely efficient metabolisms observed in many deep-sea species are direct consequences of this constant struggle for sustenance. Even with the most advanced submersibles and remotely operated vehicles, exploring and sampling this hostile environment remains an immense undertaking. The sheer logistical complexity of deploying and retrieving equipment in depths of several kilometers, the need for specialized lighting and imaging systems to pierce the absolute darkness, and the constant threat of equipment failure under extreme pressure make deep-sea research extraordinarily challenging and costly. Each sample brought to the surface must be handled with extreme care, as many deep-sea fish adapted to immense pressure can suffer severe decompression sickness as they ascend their gas-filled swim bladders, expanding fatally. The delicate gelatinous bodies of many invertebrates can be easily damaged by conventional sampling nets. Consequently, the pace of discovery is slow, painstaking, and often limited to what can be observed and collected through the narrow viewports and robotic arms of our technological envoys. For every creature successfully brought to the surface and formally described, countless others are glimpsed fleetingly, their images captured in a brief flash of light, their existence confirmed but their full story remaining untold, a silent testament to the enduring mysteries of this profoundly hostile yet infinitely fascinating deep-sea world. Taxonomy publication and the path to recognition. Even after the formidable barriers of pressure, darkness and cold have been overcome, and a specimen has been successfully brought from its abyssal home to the surface world. Its journey to formal recognition has only just begun. It enters a different kind of challenging environment, a meticulously structured realm governed by the quiet, unwavering principles of taxonomy and scientific publication. This is the formal veil, a necessary, intricate process through which a newly encountered life form transitions from a mere observation or a preserved sample into a named, recognized, and officially acknowledged species within the grand lexicon of life. Without this rigorous passage, a creature, no matter how extraordinary, remains in a liminal state, discovered, perhaps studied, but not yet fully integrated into the collective scientific understanding existing outside the widely accepted narrative of biodiversity. Taxonomy. The science of classifying and naming organisms is the very foundation upon which our understanding of life's diversity is built. It provides a universal language, a system of order, that allows scientists across the globe to communicate unambiguously about specific species. This intricate framework, largely rooted in the Linnaean system, organizes life into a hierarchical structure: kingdom, phylum, class, order, family, genus, and finally, species. Each level represents a broader grouping, a widening circle of shared characteristics, culminating in the unique designation of a single, distinct life form. When a deep-sea submersible returns with a peculiar, never-before-seen creature, the first silent question that echoes in the minds of the scientists is, where does it fit within this immense, interconnected tree of life? Is it a new branch, a new twig, or an entirely new root system waiting to be charted? The initial phase of recognition begins with the meticulous examination of the collected specimen. Imagine a delicate, almost ethereal deep-sea fish, perhaps a gulper eel larva, or a fragile gelatinous comb jelly, carefully extracted from its pressurized container, its form often distorted by the ascent. Its iridescent scales or its translucent tissues, once shimmering under the submersible's lights, now lie still under the focused beam of a laboratory microscope. Every minute detail is scrutinized: the number and arrangement of its fins, the precise morphology of its teeth, the structure of its eyes, or lack thereof, the pattern of its photophores, those light-producing organs that speak volumes about its life in darkness. Measurements are taken with painstaking precision, each millimeter recorded, each proportion calculated, building a comprehensive physical profile. This morphological analysis is often the first layer of the formal veil, a patient observational study that seeks to understand the creature's external and internal architecture. In the modern era, this traditional morphological study is increasingly complemented and often confirmed by the invisible language of genetics. Tiny tissue samples, sometimes no larger than a grain of rice, are carefully taken from the specimen. These fragments hold the very blueprint of life, the DNA, that encodes its unique identity. Through sophisticated molecular techniques, this genetic material is extracted, amplified, and sequenced. The resulting DNA sequences, often focusing on specific barcode genes that vary between species, are then compared against vast databases of known organisms. This genetic fingerprinting can reveal whether the specimen truly represents a novel species or if it is a previously described organism with unusual variations, or perhaps a larval form of a known adult. It is a powerful tool peering beyond superficial resemblances to confirm the deep evolutionary relationships that bind all life and to delineate the boundaries between distinct species with an unprecedented level of clarity. Once a specimen is tentatively identified as a new species, a crucial step in its formal recognition is the designation of a type specimen. This single physical individual becomes the permanent authoritative reference point for that species. It is carefully preserved, often in alcohol or formalin, and accessioned into a reputable natural history museum or scientific collection where it will be stored indefinitely under precisely controlled conditions. This type specimen is not merely a curiosity, it is the ultimate arbiter, the physical embodiment of the species' description. Should future scientists question the identification of a particular organism, they can always return to the type specimen, compare their findings, and resolve any ambiguities. The meticulous care taken in preserving these deep-sea types is paramount, as many are incredibly delicate. Their tissues prone to degradation or damage, making their long-term integrity a continuous challenge. With the physical and genetic evidence assembled, the most substantial part of the formal veil, the scientific description, begins. This is a rigorous, often lengthy process of writing a formal scientific paper, a document that will introduce the new species to the global scientific community. The description must be exhaustive, detailing every observable characteristic, every measurement, every genetic finding. It must meticulously compare the new species to its closest known relatives, highlighting the unique features that unequivocally distinguish it. The language used is precise, technical, and adheres to strict scientific conventions, leaving no room for ambiguity. Illustrations, whether detailed hand-drawn plates or high-resolution photographs, are essential, providing visual clarity to the textual description. This paper is not merely a report, it is an argument, a carefully constructed case for the existence and distinctness of a new form of life. Integral to this description is the act of naming the species. This follows the rules laid out in the International Code of Zoological Nomenclature, or ICZN, a universally accepted set of guidelines that ensures each species has a unique, stable, and unambiguous scientific name. The name consists of two parts: the genus name, which is always capitalized, and the species epithet, which is lowercase. Both are italicized, forming the binomial name such as Bathylogycthis profundus for a deep-sea smelt. The chosen name often reflects a distinguishing characteristic of the species, its habitat, the location of its discovery, or honors a scientist or benefactor. This act of naming, while seemingly simple, bestows upon the creature its unique identity, a label that transcends language barriers and cultural differences, allowing scientists worldwide to refer to it with absolute certainty. Once the manuscript is meticulously prepared, it embarks on the final critical stage of the formal veil publication in a peer-reviewed scientific journal. The paper is submitted to a specialized journal where it undergoes a stringent evaluation process by other experts in the field. These peer reviewers, anonymous and independent, critically assess the methodology, the scientific rigor of the analysis, the accuracy of the description, and the validity of the conclusions. They scrutinize every claim, every comparison, every piece of evidence, often suggesting revisions, requesting additional data, or even challenging the very premise of the new species designation. This peer review process, though sometimes lengthy and demanding, is the cornerstone of scientific integrity, ensuring that only robust, well-supported research enters the permanent scientific record. Only after successfully navigating this gauntlet of expert scrutiny is the paper accepted for publication. The publication itself marks the official announcement of the new species to the world. It is then that the creature, once a mere glimmer in the depths or a silent specimen in a jar, officially enters the scientific canon. Its name appears in databases, its characteristics are catalogued, and its existence is formally acknowledged by the global scientific community. From this point forward, it can be cited in research, included in biodiversity assessments, and studied by future generations of scientists. This entire process, from initial collection to final publication, can take years, even decades, especially for deep-sea organisms. The rarity of expeditions, the difficulty of obtaining high-quality specimens, the sheer complexity of the organisms themselves, and the limited number of taxonomists specializing in deep-sea life all contribute to this extended timeline. And it is within this protracted, intricate dance of discovery analysis and formalization that the concept of undisclosed deep-sea creatures truly takes root. The formal veil, while essential for scientific rigor, can inadvertently create a significant lag between the moment a creature is first encountered and the moment it receives its official name and place in the textbooks. Specimens might be collected, identified as potentially new, but then sit in museum collections for years, awaiting the focused attention of a taxonomist with the specific expertise to fully describe them. Funding for taxonomic research is often scarce, and the number of specialists capable of identifying and describing complex deep-sea fauna is dwindling. This creates a bottleneck, a backlog of potentially new species awaiting their formal introduction to the world. Furthermore, some specimens may be unusual, controversial, or damaged during collection, making their definitive description challenging. They might represent hybrid forms or individuals with extreme variations, blurring the lines of species distinction and requiring extensive debate and further evidence before a consensus can be reached. In such cases, these creatures might be known to a small circle of specialists, discussed in private correspondence, or even mentioned in conference presentations, but never formally published. They exist in a kind of scientific purgatory, known but unannounced, contributing to the whispered lore of the deep, a testament to the immense ongoing effort required to truly unveil the full, astonishing diversity of life that flourishes beneath the formal veil of our vast, enigmatic oceans. Reasons for undisclosed marine life. The journey from the abyss to the hallowed pages of a scientific journal is fraught with unseen obstacles, creating a fascinating realm where discovery precedes formal declaration. Within the quiet archives of research institutions across the hushed corridors of natural history, museums, and in the careful notes of deep-sea explorers reside glimpses of marine life known to a select few, yet held in a state of suspended animation, awaiting their official introduction to the wider world. These are the shadows of the subsurface creatures whose existence is confirmed, yet whose stories remain largely untold, held back by a complex interplay of logistical hurdles, scientific challenges, and the sheer, overwhelming scale of the unknown ocean. One of the most profound reasons for this lingering secrecy lies in the sheer rarity and ephemeral nature of deep-sea encounters. Imagine the vast, three-dimensional expanse of the deep ocean, a volume of water so immense that it dwarfs all terrestrial habitats combined. Within this colossal void, many deep-sea creatures exist at extremely low population densities, often widely dispersed across kilometers of featureless seafloor or suspended within the dark water column. To encounter a particular species, let alone collect a pristine specimen, is often a matter of immense luck, akin to finding a single specific needle in an ocean of hay. Deep-sea expeditions are extraordinarily costly, requiring specialized vessels, highly trained crews, and cutting-edge technology such as remotely operated vehicles or ROVs, and manned submersibles, each dive representing a monumental investment of resources and time. These opportunities are infrequent, precious windows into a hidden world, and each encounter with a potentially new species is a fleeting moment often impossible to replicate. A creature might flash past a camera or be caught in a trawl net only once, yielding a single, often damaged specimen. This singular nature of discovery makes it incredibly difficult to gather the multiple specimens needed to fully understand species variation, and thus to confidently describe a new species according to rigorous scientific standards. Furthermore, the very act of bringing these delicate denizens of the deep to the surface presents its own set of formidable challenges. Many deep-sea organisms, particularly those inhabiting the bathypelagic and abyssal zones, are exquisitely adapted to immense pressure and near-freezing temperatures. Their bodies are often soft, gelatinous, and highly fragile, composed largely of water and incompressible tissues designed to maintain hydrostatic equilibrium with their surroundings. The rapid ascent through kilometers of water to the surface can be catastrophic. Changes in pressure can cause internal organs to rupture swim bladders, to expand explosively, and delicate tissues to tear. What might have been a vibrant, intact creature in its natural habitat can arrive at the surface as a mangled, unrecognizable mass, its intricate structures utterly destroyed by the journey. Even with specialized pressure-retaining samplers which attempt to maintain ambient pressure during ascent. The delicate nature of these organisms makes preservation difficult. Their colors, often vibrant and specific in the abyss, can fade instantly upon exposure to surface light and oxygen, or simply dissolve into the preserving fluid, obscuring crucial morphological details. A specimen once captured might become an enigma in a jar its original beauty, and defining features lost, making accurate description a painstaking, often impossible task. Another significant factor contributing to the backlog of undisclosed deep-sea species is the taxonomic bottleneck. The global scientific community faces a dwindling number of trained taxonomists, particularly those specializing in the obscure and often challenging groups of deep-sea fauna. Taxonomy, the fundamental science of describing and classifying life, is a highly specialized field, requiring years of dedicated training and an encyclopedic knowledge of specific organismal groups. Identifying a new deep-sea amphipod, for instance, requires an expert in deep-sea amphipods who can meticulously compare the specimen's minute morphological features against hundreds, if not thousands, of known species. These experts are few and far between, often burdened with vast collections of unidentified specimens, each awaiting their focused attention. Funding for purely taxonomic research is also often scarce, as grant agencies tend to favor studies with more immediate ecological or biotechnological applications. Consequently, countless specimens carefully collected and preserved can sit for decades in museum collections, patiently awaiting the rare taxonomist with the time, expertise, and resources to formally describe them. They are known to exist catalogued by a collection number, but their scientific name, their place in the tree of life, remains an unwritten chapter. The incompleteness of collected data also plays a critical role. For a new species to be formally described and published, scientists typically require a comprehensive dataset, including detailed morphological measurements, anatomical observations, and increasingly genetic sequences. However, deep-sea sampling often yields only fragmented data. Sometimes only a single individual is collected, leaving questions about intraspecific variation. Other times, only a larval or juvenile form is captured, making it impossible to confidently link it to an unknown adult or to fully characterize the species' life cycle. Without the full suite of developmental stages or multiple adult specimens, a definitive description can be elusive. Furthermore, the environment itself conspires against comprehensive data collection. Observing behavior in the deep is notoriously difficult. Creatures are often filmed for mere seconds, their natural interactions obscured by the submersible's lights or the disturbance of sampling equipment. Without robust ecological context, the story of a new species remains incomplete, a silent whisper rather than a clear voice. Then there are the controversial or ambiguous creatures that defy easy categorization and spark intense debate among the few specialists qualified to assess them. Imagine a specimen that exhibits characteristics of two different genera, or one that possesses an extreme morphological variation that blurs the lines of established species boundaries. Such anomalies can challenge existing phylogenetic frameworks, prompting questions about hybridization, convergent evolution, or even the fundamental definitions of species themselves. In these instances, the path to formal description becomes a protracted journey through scientific discourse, requiring additional evidence, further sampling, which is often impossible, and a consensus among experts that can take years to form. These organisms might be discussed at conferences, mentioned in informal communications, or even published as species inquirenda, species of doubtful identity. But they fail to achieve the definitive, widely accepted recognition of a formally described species. They exist in a state of scientific limbo, a testament to the complex and sometimes contentious nature of biological classification. Finally, there are sometimes strategic or ethical considerations that lead to a deliberate delay in announcing a deep-sea discovery. In some rare instances, scientists might choose to temporarily withhold the formal publication of a new species or the precise location of its discovery to protect a particularly vulnerable population or a unique sensitive habitat from potential exploitation or disturbance. For example, the discovery of a new species around a hydrothermal vent system, an oasis of life in the deep, might prompt concerns that public knowledge could lead to unwanted attention from resource extraction industries or unregulated tourism, potentially jeopardizing the delicate ecosystem before adequate conservation measures can be put in place. In these cases, the shadow cast over the species is a conscious protective act, prioritizing the long-term survival of the organism and its habitat over the immediate gratification of scientific announcement. Thus, the realm of undisclosed deep-sea marine life is not a simple void of ignorance, but rather a complex landscape shaped by the immense scale of the ocean, the fragility of its inhabitants, the limitations of human technology, and the intricate demands of scientific rigor. These creatures, glimpsed but not yet fully named, represent the silent majority of deep-sea biodiversity, a constant reminder of how much remains unknown in our own planet's most vast and mysterious domain. They are the quiet inhabitants of the shadows, waiting patiently for their moment to emerge from the depths of scientific archives and take their rightful place in the grand narrative of life on Earth. Historical encounters and unconfirmed sightings. Long before the age of modern submersibles and remotely operated vehicles, when the deep ocean remained an almost entirely impenetrable mystery, whispers of unseen life began to surface, carried by the crude nets of early dredges and the awestruck accounts of those who dared to peer into the abyssal gloom. For centuries, the prevailing scientific consensus held that the deep sea beyond a few hundred meters was an abiotic wasteland, a barren, lifeless expanse utterly inhospitable to all but the most primitive forms of existence. The crushing pressure, the perpetual darkness, and the frigid temperatures were believed to render life impossible. Yet even in those nascent years of oceanic exploration, faint glimmers of contradictory evidence would occasionally emerge, hinting at a vibrant hidden world far beneath the waves, fueling speculation and igniting the scientific imagination. One of the earliest and most profound shifts in this understanding came with the epochal Challenger expedition, which embarked on its monumental circumnavigation of the globe from 1872 to 1876. This ambitious scientific voyage aboard a converted British naval corvette was specifically tasked with exploring the deep sea, systematically sampling its depths with an array of dredging equipment and sounding lines. With each deployment of their cumbersome trawls and nets brought up from kilometers below the surface, the scientists aboard the Challenger were met with an astonishing parade of life forms, many utterly alien to anything previously known. From bizarre elongated fish with luminous lures to delicate glass-like sponges and strange stalked crinoids, the sheer volume and diversity of specimens shattered the prevailing notion of an abiotic abyss. The expedition alone described over 4,700 new species, fundamentally reshaping humanity's understanding of marine biodiversity. However, even amidst this explosion of discovery, the limitations of the era meant that countless other creatures glimpsed or collected might have slipped through the cracks of formal recognition. The methods of collection were rudimentary. Heavy dredges scraped indiscriminately across the sea floor, and trawl nets, often dragged for hours through immense volumes of water, would bring up their catch from varying depths, making precise depth localization challenging. The sheer force of these operations, combined with the rapid ascent, meant that many delicate deep-sea organisms arrived on deck in a severely damaged, fragmented, or even unrecognizable state. A fragile, gelatinous creature, perfectly intact in the deep, might be reduced to an amorphous blob. By the time it reached the surface, its distinguishing features obliterated. Imagine the challenge of describing a new species from a collection of torn tissues and broken appendages, with no clear understanding of its original form or color. These damaged specimens, even if recognized as potentially new, often lacked the integrity required for a rigorous publishable description, relegating them to the unconfirmed or undescribed categories within the expedition's vast, burgeoning collections. Beyond the Challenger's formal collections, the early 20th century saw the emergence of a new breed of deep-sea explorer individuals who ventured directly into the abyss within the confines of pioneering submersibles. One of the most famous of these early ventures was the bathysphere, designed by Otis Barton and piloted by William Beebe. In the 1930s, these two intrepid explorers made a series of descents off the coast of Bermuda, reaching depths of over 900 meters. Their sphere, a cramped steel chamber with thick quartz windows, offered the first direct human eyes into the mesopelagic and upper bathypelagic zones. What Beebe described in his vivid accounts were not always formally collected specimens, but breathtaking, fleeting visual encounters. He spoke of large, vague forms passing by the viewport of serpent-like creatures with glowing patterns and of great luminous eyes peering back from the blackness. While his observations were meticulous and detailed, they were, by their very nature, unconfirmed sightings. Without a specimen to retrieve, without a clear photograph to analyze, these magnificent creatures remained in the realm of anecdotal evidence whispers, from the deep yet powerfully suggestive of a far greater diversity than even the Challenger had revealed. These unconfirmed sightings and fragmented collections contributed to a rich tradition of sea monster lore, where the truly bizarre and enormous denizens of the deep often blurred the line between myth and reality. For centuries, tales of colossal serpents, multi-tentacled beasts, and giant fish circulated among sailors and coastal communities. While many of these were undoubtedly exaggerations or misidentifications of known animals, some were almost certainly rooted in rare, fleeting encounters with genuine deep-sea leviathans. The giant squid Architeuthis dux stands as a quintessential example. For centuries, its existence was considered mythical, a creature of legend known only through the massive scars found on sperm whales and the occasional fragmented carcass washed ashore. It was not until the late 19th and early 20th centuries that intact specimens began to be examined, slowly pulling the giant squid from the realm of folklore into confirmed zoological fact. Even so, the first photographs of a living giant squid in its natural habitat were not captured until 2004, and clear video footage not until 2012. This extended journey from legend to documented reality for Architeuthis strongly suggests that other truly colossal and as yet undescribed deep-sea organisms might still lurk in the ocean's vast, unexplored reaches, occasionally glimpsed, but rarely, if ever, formally captured or identified. Beyond the giant squid, other cryptids, creatures whose existence is unproven but widely believed, also find their possible origins in deep-sea sightings. The legendary kraken, for instance, might be an amalgamation of giant squid and other large deep-sea cephalopods, the globsters. Those mysterious amorphous carcasses that occasionally wash ashore, defying identification, could be the remains of unknown large deep-sea organisms whose bodies undergo extreme decomposition or unusual physical changes upon reaching the surface. Scientists examining these globsters often struggle to definitively classify them, leaving their origin shrouded in mystery and fueling speculation about colossal, undiscovered forms of life in the deep. Each such incident, whether a fleeting visual or a perplexing carcass, adds another layer to the whispers from the deep past, reminding us that our knowledge is far from complete. Even with the advent of early deep-sea cameras and remotely operated vehicles in the mid to late 20th century, the challenges of documentation persisted. Early cameras, with their limited resolution and often jerky movements, would capture fleeting images of unknown creatures in the beam of their artificial lights. A strange, elongated shape might drift through the frame, or a peculiar glowing organism might briefly appear before vanishing into the darkness. These were often too indistinct, too brief, or too poorly illuminated to allow for definitive identification or formal description. They became ghost sightings in the photographic archives, compelling yet unconfirmable evidence of life beyond our current understanding. These early visual records, while not leading to formal species descriptions, served as tantalizing hints, confirming the existence of bizarre forms that could not be adequately sampled or studied at the time. The issue of lost samples and inadequate preservation also contributes to the enduring mystery of unconfirmed deep-sea species. In the early days of oceanographic research, preservation techniques were not as refined as they are today. Delicate specimens might have deteriorated rapidly during transit back to shore, or been improperly stored in collections, leading to their eventual loss or decay beyond recognition. A unique fish collected on a pioneering expedition a century ago might have been carefully documented in field notes, but if the physical specimen itself was compromised, its formal description could never be completed. These lost samples represent invaluable fragments of biodiversity, now forever beyond our reach, their potential for discovery tragically unfulfilled. The scientific community has become far more meticulous in its collection and preservation protocols, but the ghosts of these lost specimens continue to haunt the historical record, silent testaments to discoveries that almost were. Finally, the anecdotal evidence from fishermen and commercial trawlers, while rarely considered scientifically robust, occasionally offers intriguing glimpses into the unknown. Deep-sea fishing operations, particularly those using bottom trawls, can inadvertently bring up creatures from extreme depths that are entirely unexpected. A fisherman might report a bizarre, colossal, squid-like creature caught in his nets, or a strange luminescent fish unlike anything he has ever seen. While these accounts are often difficult to verify and the specimens themselves are usually discarded, they sometimes contain kernels of truth, aligning with what scientists know of deep-sea adaptations and the potential for immense undiscovered life forms. These stories, passed down through generations of those who worked the seas, form a kind of informal deep-sea folklore, a continuous stream of whispers from the deep, reminding us that the ocean's greatest secrets may still reside just beyond the reach of our most advanced scientific instruments, patiently waiting for their moment to emerge from the profound, abyssal silence. Controversial and disputed findings. Within the silent, cool confines of the abyssal archives, amidst rows of glass jars and meticulously labeled vials, reside specimens that whisper tales of profound scientific disagreement and unresolved mystery. These are the anomalies, the controversial findings, the disputed creatures whose very existence or classification has sparked protracted debates among the few scientists privileged to study them. Unlike the smoothly catalogued species that gracefully find their place in the tree of life, these organisms linger in a state of taxonomic limbo. Their identities questioned, their relationships contested, their stories perpetually unfolding under the critical gaze of expert scrutiny. They represent the frontier where scientific consensus wavers, where the boundaries of knowledge are pushed, and where the deep ocean continues to challenge our very frameworks of understanding. The intrinsic difficulties of deep-sea exploration themselves lay fertile ground for these controversies. Imagine a research team, after months of preparation and a perilous descent, retrieves a unique specimen from a depth of 7,000 meters. The creature, perhaps a fragile gelatinous organism with an unusual arrangement of bioluminescent organs, is brought to the surface inevitably traumatized by the ascent. Its delicate structures may be partially damaged, its original vibrant colors faded, its precise form altered. A single compromised specimen, no matter how extraordinary, immediately presents a challenge to definitive classification. Is it a new species, or merely a highly aberrant individual of a known species, its morphology distorted by environmental stress or the trauma of capture? Without multiple specimens, without observing it in its undisturbed natural habitat, and without a comprehensive understanding of its life cycle, certainty remains elusive opening the door for differing interpretations and prolonged scientific discussion. One common source of dispute arises from the phenomenon of morphological ambiguity. Deep-sea environments with their extreme and stable conditions often drive convergent evolution, where unrelated species develop similar physical traits in response to similar selective pressures. A fish from one family might evolve a flattened body and upward-pointing eyes mirroring a completely unrelated species from a different lineage, both adapted for life on the abyssal plane. Distinguishing between genuine evolutionary relatedness and superficial resemblance becomes a painstaking task, requiring meticulous examination of internal anatomy, skeletal structures, and often the invisible language of genetics. Conversely, some deep-sea species exhibit remarkable polymorphism, where individuals within the same species can display significant variations in size, color, or even the presence of certain appendages depending on their age, sex, or specific microhabitat. A taxonomist encountering two such variants might initially classify them as distinct species, only for later research, perhaps with genetic sequencing, to reveal their shared identity, leading to a re-evaluation and consolidation of previous findings. These cases highlight the subtle yet profound challenges of visual identification in a realm where life takes on such incredibly diverse and often counterintuitive forms. The advent of molecular genetics, while revolutionizing taxonomy, has also introduced its own set of controversies, particularly in the realm of cryptic species. These are morphologically indistinguishable species that are in fact genetically distinct, having diverged evolutionarily despite retaining identical or near-identical external appearances. In the vast, dark, and often uniform environment of the deep sea, where visual cues are less critical, cryptic speciation is believed to be particularly common. A deep-sea worm, for example, might appear identical to a known species under a microscope, but its DNA might reveal a genetic divergence significant enough to warrant its classification as an entirely new species, one that has been living unseen, unannounced alongside its morphological twin for millennia. Such discoveries can be highly contentious, challenging long-held morphological classifications and forcing scientists to re-examine entire genera or families, leading to heated debates about the precise definition of a species and the relative weight given to morphological versus genetic evidence in deep-sea environments. Perhaps one of the most celebrated and initially controversial deep-sea discoveries, though not strictly an undisclosed species in the modern sense, is the coelacanth. Its story serves as a powerful illustration of how a scientific finding can shatter established paradigms and ignite fierce debate. Believed to have gone extinct with the dinosaurs some 66 million years ago, this ancient lobe-finned fish was known only from fossil records. Then, in 1938, a living specimen was caught off the coast of South Africa. The initial announcement was met with a mixture of awe and skepticism. How could such a large, distinctive fish have evaded detection for so long? Was it a genuine, living fossil or a misidentification? The subsequent discovery of a second species, Latimeria menadoensis, in the waters off Indonesia in 1997, further fueled scientific excitement and debate, confirming the existence of a truly ancient lineage that had persisted largely unchanged in the deep, quiet waters of the Indian Ocean. The coelacanth remains a touchstone for discussions about relict species and the potential for other lost lineages to persist in the unexplored depths, reminding scientists to always question assumptions about extinction. Beyond living fossils, the discovery of entirely new body plans or evolutionary lineages can also lead to significant controversy. When the first hydrothermal vent ecosystems were discovered in 1977, the very concept of life fueled by chemosynthesis rather than photosynthesis was revolutionary. The bizarre tube-dwelling giant tube worms, Riftia pachyptila, which lack mouths and digestive tracts, instead relying on symbiotic bacteria for sustenance, challenged fundamental biological principles. Their classification, their evolutionary relationships, and even their basic physiology sparked intense debate, pushing the boundaries of what was considered possible for multicellular life. These were not just new species, they were new ways of being alive, forcing a re-evaluation of the very definition of an ecosystem and the origins of life itself. Even decades later, new vent and seep species continue to be discovered, each one adding to the complexity and sometimes controversy of these unique, deep-sea biological communities. The very act of naming a new species can also become a point of contention, particularly in highly competitive fields, or when there are multiple research groups working on similar material. The rules of nomenclature dictate that the first validly published name takes precedence. However, if two research teams are simultaneously describing the same new species, or if there is a dispute over who first collected or identified a specimen, the process can become entangled in priority disputes. Such disagreements, while perhaps appearing trivial from an external perspective, are deeply significant within the scientific community, impacting authorship recognition and the historical record of discovery. These priority disputes can delay formal publication or lead to later reclassifications, leaving a species' official name in flux for a period. Furthermore, some organisms exist in the abyssal archives as species inserte sedis or species inquirenda. These Latin terms denote species of uncertain placement or doubtful identity. A specimen might be collected that is clearly unique, clearly distinct from anything known, but it might be too damaged, too incomplete, or too anomalous to be confidently placed within an existing genus or family. It sits in the collection, a silent enigma waiting for more complete material or for new analytical techniques, or for the insights of a future taxonomist to definitively resolve its identity. These are the truly undisclosed species in the most profound sense. Their existence is known, their uniqueness recognized, but their formal scientific story remains unwritten, held back by the sheer ambiguity of their form or the incompleteness of the available evidence. They are the spectral entities of the deep-sea catalogue, known but not fully understood, a constant reminder of the limits of our current knowledge. The role of advanced imaging technologies, while immensely beneficial, can also contribute to these ongoing disputes. High-resolution cameras on ROVs and submersibles capture stunning images and videos of deep-sea life in its natural habitat, offering unprecedented insights into behavior coloration and morphology. However, these visual records, while invaluable, are not always sufficient for formal species description, which typically requires a physical specimen for detailed examination and preservation. An ROV might capture breathtaking footage of a large unknown squid species or a unique glowing jellyfish, but without a specimen to collect it, remains an unconfirmed sighting, albeit a highly compelling one. The debate then shifts to the validity of photographic evidence versus physical specimens for species delineation, particularly for rare and elusive creatures that may never be captured. This often leads to a creature being widely known among specialists through video, but still lacking a formal scientific name existing in the shadows of the subsurface, a visual anomaly yet to be fully embraced by the formal taxonomic process. Ultimately, the controversial and disputed findings in the abyssal archives are not failures of science, but rather testament to its dynamic and self-correcting nature. they underscore the immense challenge of exploring and classifying life in the planet's largest, least-known habitat. Each anomaly, each debate, each re-evaluation pushes scientists to refine their methods, to question their assumptions, and to delve deeper into the intricate evolutionary pathways that have shaped life in the perpetual darkness. These lingering mysteries, these unresolved classifications, serve as a potent reminder that the deep ocean continues to hold countless secrets, waiting patiently for the patient, persistent work of scientists to lift the formal veil and bring these extraordinary, often disputed forms of life into the full, clear light of understanding. They are the silent, enigmatic questions posed by the deep, inviting us to listen more closely to their whispers and to ponder the vastness of what remains unknown beneath the waves. The tale of disappearing samples and forgotten data. Even as modern science strives for meticulous documentation and preservation, the deep abyssal archives hold not only controversies, but also the poignant echoes of what has been lost. Within the vast, silent repositories of scientific history, there are tales of specimens once glimpsed, once collected, once held in the hand of a hopeful scientist, only to vanish, leaving behind mere whispers of their existence. This is the narrative of disappearing samples and forgotten data, a profound loss that directly contributes to the vast realm of undisclosed deep-sea life, creating gaps in our understanding that may never be filled. Each lost specimen, each misplaced note, represents a potential species forever relegated to the realm of the unknown, a silent testament to the fragility of discovery itself. The initial moment of loss often begins at the very point of collection in the deep ocean. As we have slowly descended through the preceding chapters, the extreme conditions of pressure, darkness, and cold have presented formidable challenges to life itself. They also conspire against the successful retrieval of intact specimens. Many deep-sea organisms, particularly those from the bathy and hadal pelagic zones, are incredibly delicate. Their bodies often soft, gelatinous, or extremely fragile, built to yield to the immense pressure rather than resist it. Imagine a gossamer-thin jellyfish, its bell spanning meters, its intricate tentacles trailing through the water like strands of fine silk. Such a creature, when caught in a trawl net or even carefully manipulated by an ROV's robotic arm, can be easily torn, crushed, or dismembered by the sheer forces involved in its capture and ascent. A specimen that appears whole on an underwater camera might arrive on deck as a fragmented, unrecognizable mass, its defining features obliterated, rendering it useless for formal description. The dream of a new species dissolves into a collection of unidentifiable biological debris. In the nascent years of deep-sea exploration, the challenges were even more pronounced. Early dredges and nets, while revolutionary for their time, were crude instruments indiscriminately scraping the sea floor or dragging through vast water columns. The organisms collected often suffered immense trauma, arriving in a state of disrepair that made detailed morphological analysis nearly impossible. Furthermore, the understanding of deep-sea biology and preservation techniques was rudimentary. Scientists were often working on ships far from shore, with limited resources and knowledge of how best to stabilize and preserve these unique life forms. Many delicate tissues would simply degrade rapidly upon exposure to surface oxygen and light, or in inadequate preserving fluids. Colors which might have been crucial for identification or understanding bioluminescent patterns would fade or leach away, leaving behind a monochrome ghost of the original creature. A unique, iridescent fish, once a shimmering jewel in the darkness, might become a dull, colorless form in a jar. Its key characteristics lost to the passage of time and the limitations of early preservation. Beyond the initial trauma of collection and the challenges of early preservation, the sheer volume of material brought back from large expeditions often overwhelmed the capacity of museums and research institutions. Historically, expeditions like the Challenger brought back tens of thousands of specimens, representing countless hours of painstaking collection. The task of sorting, identifying, and cataloguing this immense bounty fell to a relatively small number of taxonomists, who often worked for years, even decades, on the material. In this deluge of new life, it was inevitable that some specimens, perhaps less striking or more damaged, would be set aside temporarily shelved or simply lost in the vastness of an expanding collection. Imagine a back room in a grand old natural history museum, filled with shelves stretching to the ceiling, laden with jars and bottles, each containing a silent mystery from the deep. Amidst this overwhelming abundance, a small, unremarkable vial might be misplaced. Its label smudged, its contents slowly deteriorating, its potential for discovery fading into obscurity. The problem of lost samples extends beyond physical misplacement. Over time, the very integrity of preserved specimens can diminish. Preservatives like alcohol can evaporate if seals are not perfectly maintained, leading to desiccation and decay. Formalin, a common fixative, can become acidic over decades, slowly dissolving calcium carbonate structures like shells or bones, or altering soft tissues beyond recognition. Even carefully stored specimens, if not regularly monitored and replenished with fresh preserving fluids, can suffer irreversible damage. A unique deep-sea worm, once a pristine type specimen, might, after a century of storage, become a fragile, amorphous mass, its defining features eroded by the relentless chemistry of time and inadequate care. Such degradation means that even if a specimen is physically present, its scientific utility, its ability to definitively represent a new species, can be irrevocably compromised, turning a once clear discovery into an ambiguous echo. Crucially, the loss of a specimen is often compounded by the disappearance of its associated data. A specimen, no matter how perfectly preserved, is only as valuable as the information that accompanies it. This includes precise collection locality, latitude, longitude, depth, date of collection, method of capture, environmental parameters, temperature, salinity, and detailed field notes describing its appearance, color, and behavior in life. In the past, this data was meticulously recorded in paper logbooks, notebooks, and on specimen labels. These physical records are vulnerable to fire flood pests or simply the slow process of degradation. A single misfiled page, a faded ink entry, or a lost notebook can render a specimen scientifically worthless, severing its connection to the precise moment and place of its discovery. Without this crucial contextual information, a unique deep-sea fish in a jar becomes an orphan, its story untold, its scientific value severely diminished, even if its physical form endures. The human element also plays a significant role in the tale of forgotten data. Scientific careers are often long and dedicated, with researchers accumulating vast amounts of specialized knowledge, particularly about their specific collections. When a senior taxonomist retires or passes away, their unique expertise, their intimate familiarity with specific specimens, and their accompanying notes can be lost. Institutional memory, the collective knowledge held within a research group or museum, can erode over time, especially if there isn't a direct successor to continue the work. A collection of undescribed deep-sea sponges known only to one particular expert might languish for decades if that expert's knowledge is not formally transferred and disseminated. The unique insights, the tentative identifications, the half-formed hypotheses that reside in a researcher's mind or in their personal unpublished notes can simply evaporate, taking with them the potential for numerous new species to be formally recognized. The shift in scientific priorities over time can also contribute to the neglect of historical collections. While taxonomy remains fundamental, the emphasis in marine science has shifted towards ecological studies, conservation biology, and molecular genetics. Funding for purely descriptive taxonomic work, especially on older, already collected material, can be challenging to secure. As a result, new generations of scientists may focus on fresh data and contemporary expeditions, leaving vast historical collections unexamined, their secrets still locked within the jars and the faded handwriting of past explorers. These collections, brimming with potential new species, become silent archives, their contents waiting for a resurgence of interest, a new generation of taxonomists, or perhaps for new technologies that can extract information from even compromised specimens. The digital age, while offering solutions, also introduces its own set of vulnerabilities. While modern data is often digitized and stored in vast databases, these digital files are not immune to loss. Corrupted hard drives, obsolete file formats, and the continuous evolution of data management systems can lead to digital decay, where information becomes inaccessible or unreadable over time. A meticulously scanned image of a unique deep-sea organism or a comprehensive genetic sequence could one day be lost to technological obsolescence, becoming a digital echo, just as elusive as a faded paper record. The promise of perpetual digital preservation is a powerful one, but it requires continuous vigilance, migration, and maintenance, a daunting task for institutions managing petabytes of diverse scientific data. Ultimately, the echoes of the lost, the disappearing samples and forgotten data represent a profound underestimation of deep-sea biodiversity. Each lost specimen is not just a physical object, it is a lost opportunity to name a new species, to understand a unique adaptation, to trace an evolutionary lineage, or to contribute to the grand mosaic of life on Earth. These silent losses mean that our current understanding of deep-sea diversity, vast as it is, remains incomplete, a mere shadow of the true richness that exists beneath the waves. The ghost of a uniquely adapted creature glimpsed once and then lost to the ravages of time or circumstance continues to haunt the scientific imagination, a poignant reminder of the immense, ongoing challenges of unveiling the deep ocean's true biological wealth. They are the untold stories, the unwritten chapters lingering in the quiet depths of our collective scientific memory, waiting for a chance, however slim, to finally emerge from the profound, silent archives of the unknown. Hints of unseen leviathans. As our gaze drifts deeper into the profound, unlit expanse of the ocean, a different kind of mystery begins to coalesce, one that resonates with ancient human fears and tantalizing hopes. The possibility of truly colossal, undiscovered life forms. Beneath the vast, aqueous veils in the crushing silence of the abyss, scientists and explorers occasionally encounter hints, mere fleeting suggestions of leviathans far grander and more enigmatic than anything yet formally catalogued. These are the giants beneath the veils, creatures that persist in the realm of speculation, their existence hinted at by indirect evidence, unconfirmed sightings, or the sheer, overwhelming scale of the unexplored deep. The human imagination has always been drawn to the colossal, to the idea of something immense and powerful lurking just beyond our perception, and in the ocean's depths, this primal fascination finds its most fertile ground. For centuries, tales of sea monsters, the kraken, the great sea serpent, the monstrous leviathan, have woven themselves into the fabric of maritime folklore. While many such accounts were undoubtedly born from exaggeration, fear, or misidentification of known animals, some were almost certainly rooted in genuine, albeit rare encounters with extraordinarily large deep-sea organisms. The most compelling example of this transition from myth to scientific fact is the giant squid Architeuthis ducks. For generations, its existence was relegated to the realm of sailors' yarns, a creature of legend known only through fragmented carcasses washed ashore or the massive circular scars found on the bodies of sperm whales. It took immense scientific effort spanning centuries to gather enough physical evidence intact specimens, detailed anatomical studies to formally describe and acknowledge this magnificent cephalopod. Even then, the first photographs of a living giant squid in its natural habitat were not captured until 2004, and clear video footage not until 2012, illustrating just how elusive even a 15-meter-long animal can be in the deep ocean. The protracted journey of Architeuthis from myth to documented reality serves as a powerful reminder that some of the most fantastical legends might indeed hold kernels of truth, suggesting that other, truly colossal as yet undescribed deep-sea organisms might still lurk patiently waiting for their moment of revelation. Beyond the giant squid, other forms of indirect evidence periodically surface, hinting at the presence of even larger, unknown predators. The very bodies of sperm whales, those deep-diving behemoths of the ocean, often bear scars from epic battles with their cephalopod prey. While many of these scars are consistent with known species like the giant squid, some are far larger, or of an unusual shape suggesting encounters with truly gargantuan, currently undescribed cephalopods. Perhaps even a colossal giant squid that dwarfs Architeuthis. Scientists theorize that these whales capable of diving to depths of over 2,000 meters may be encountering species that reside in realms far below our current reach. Creatures so immense and powerful that their existence is only inferred through the wounds they inflict upon their equally colossal predators. The silent, scarred testimony on a whale's flank becomes a cryptic map to an unseen world of titanic struggle. Periodically, the ocean also yields its mysteries in the form of globsters, amorphous, often grotesque masses of organic material that wash ashore, defying immediate identification. These perplexing carcasses are often subjected to intense scrutiny, yet their origin frequently remains elusive. While many globsters are eventually identified as decomposed remains of known large marine animals, such as whales or sharks, a significant number resist classification. Their tissues so altered by decomposition, or their morphology so unusual that they cannot be definitively linked to any known species. Scientists speculate that some of these enigmatic masses could potentially be the remains of truly giant, undiscovered deep-sea creatures whose unique biology and immense size lead to unusual decomposition patterns upon reaching the surface. The deep-sea environment, with its cold temperatures and immense pressure, can preserve tissues in ways unlike surface conditions, and the subsequent rapid ascent and exposure to warmer, oxygen-rich waters could cause dramatic physical changes, transforming an unknown leviathan into an unrecognizable enigma. Each globster therefore becomes a silent question mark, a physical hint of life forms that exist beyond the current boundaries of our scientific understanding. Acoustic anomalies from the deep ocean also offer tantalizing, albeit indirect, evidence of unseen giants. The ocean is a realm of sound, and hydrophones deployed across the globe have recorded a fascinating array of biological and geological noises. Among these, certain ultra-low frequency sounds have occasionally been detected that defy easy explanation. One famous example is the bloop, a powerful ultra-low frequency sound detected by the US National Oceanic and Atmospheric Administration, or NOAA, in 1997. The sound's characteristics suggested an animal source, yet its immense volume and duration were far greater than any known creature, even the largest whales. While later analysis tentatively attributed the bloop to icequakes, the sound of icebergs cracking and breaking, the initial mystery underscored the potential for truly colossal vocalizing organisms to exist in the deep. Scientists continue to monitor the deep-sea soundscape, ever vigilant for unusual acoustic signatures that might hint at the presence of immense, undiscovered life forms navigating the vast, dark ocean. The sheer scale of the deep, where sound travels thousands of kilometers, means that a creature could be hundreds or thousands of kilometers away, yet still register its presence through an enigmatic acoustic signature. The very ecology of the deep sea supports the theoretical existence of such giants through the phenomenon known as deep-sea gigantism. This is a well-documented biological trend where certain animal groups, when found in deep-sea environments, evolve to be significantly larger than their shallower water relatives. Examples include giant isopods, giant amphipods, and the colossal squid Mesonychoteuthis hamiltoni, which is generally considered larger in mass than the giant squid Architeuthis dux, though perhaps not in total length. Several theories attempt to explain this phenomenon. One suggests that the extreme cold of the deep ocean leads to slower metabolic rates, resulting in extended lifespans and continuous growth over a longer period. Another theory posits that the scarcity of food resources in the deep favors larger body sizes, as larger animals can store more energy reserves, withstand longer periods between meals, and are more efficient at foraging across vast, sparse areas. Furthermore, the absence of visual predators in the perpetual darkness may reduce the selective pressure against large body size. These ecological drivers create an environment uniquely conducive to the evolution of immense, slow-growing and long-lived creatures, making the concept of undiscovered deep-sea giants scientifically plausible. Direct visual encounters, though exceedingly rare, also contribute to the whispers of unseen leviathans. Submersibles and remotely operated vehicles during their brief forays into the abyssal depths occasionally capture fleeting glimpses of creatures that defy identification. A massive, indistinct shadow might pass through the limited beam of a submersible's lights too quickly to be properly identified or photographed. An ROV camera might capture a brief out-of-focus image of an unusually large unknown fish or invertebrate, its true form obscured by the murky water and the limitations of deep-sea optics. These are not formal discoveries, but rather tantalizing hints, visual fragments that leave scientists pondering the true scale and diversity of life that remains hidden. Imagine the quiet awe of a scientist watching a screen, seeing a shape, a movement, a hint of something vast and unknown, only for it to vanish back into the impenetrable darkness, leaving behind only a lingering sense of wonder and a renewed conviction that the deep still holds its grandest secrets. Deep-sea trawling operations, particularly those targeting fish at depths of several hundred to a few thousand meters, sometimes bring up accidental bycatch that sparks immediate scientific interest. While most of these are known species, there are occasional instances where unusually large or entirely unknown specimens are retrieved. These might be enormous deep-dwelling sharks, far larger than any formerly described, or colossal fish, with bizarre anatomies that defy current classification. Often these specimens are damaged by the trawling process or are simply discarded by commercial fishermen before scientists can properly examine them. However, the rare instances where such unusual captures are brought to scientific attention provide concrete, albeit anecdotal, evidence that the deep-sea fishing industry, in its indiscriminate sweep, occasionally encounters creatures that remain outside the established scientific record, reinforcing the idea of a hidden population of giants. The very vastness of the deep ocean, covering more than half of our planet's surface and representing the largest contiguous habitat on Earth, provides an immense canvas for the existence of undiscovered leviathans. Even with decades of exploration, only a minuscule fraction of the deep sea has been directly observed or sampled. Imagine trying to find a specific rare animal in an area larger than all the continents, combined perpetually dark and three-dimensional. The odds of encountering a rare large creature are astronomically low. Furthermore, many deep-sea giants may be incredibly elusive, possessing behaviors that allow them to actively avoid human technology, or inhabiting specific hard-to-reach niches such as the deepest parts of abyssal plains or the unexplored reaches of submarine canyons. The veil that hides them is not just the darkness or the pressure, but the sheer overwhelming scale of their domain. A realm where even the largest beings can remain utterly invisible to our limited probes. Thus, the hints of unseen leviathans continue to echo through the scientific community, fueling both rigorous research and a profound sense of wonder. From the ancient scars on a whale's skin to the enigmatic blobs on a distant shore, from the mysterious sounds in the deep to the fleeting shadows in a submersible's lights, the evidence, though indirect, steadily accumulates. It suggests that our current understanding of deep-sea biodiversity, especially concerning its largest inhabitants, is far from complete. The deep ocean, in its quiet, dark immensity, continues to guard its greatest secrets, holding within its liquid embrace the potential for discoveries that could once again redefine our perception of life on Earth, reminding us that the greatest giants may still be waiting, patiently beneath the profound and silent veils of the unknown. Expanding our view, deepening the mystery. For countless millennia, the deep ocean remained a realm of impenetrable mystery, its colossal volume shielded from human eyes by insurmountable pressure and absolute darkness. Our understanding of its life forms was largely confined to fragmented remains washed ashore, or the occasional damaged specimen inadvertently hauled up by early fishing nets. It was a world imagined more than seen, populated by the whispers of ancient mariners and the tantalizing hints of natural philosophers. However, with the relentless march of human ingenuity, new technological eyes have slowly, painstakingly begun to pierce this profound veil, extending our senses into the abyss and allowing us to glimpse the wonders that lie beneath. Yet, with each expansion of our view, with every new creature revealed, the very act of discovery paradoxically deepens the mystery, unveiling the true staggering scale of what remains unknown. The first true technological extensions of our senses into the deep came not with sophisticated machines, but with the simplest of tools, the sounding line and the dredge. The sounding line, a weighted rope lowered into the depths, allowed early oceanographers to measure the ocean's floor, mapping its unseen topography meter by painstaking meter. The dredge, a sturdy net or basket dragged across the seafloor, brought up the first tangible evidence of life beyond the sunlit surface, shattering the long-held belief in an abiotic abyss. These rudimentary instruments, though crude, laid the foundational understanding of the deep's physical structure and hinted at its biological richness. They were the first tentative probes into a world utterly alien, confirming that even in the crushing darkness, life persisted tenacious and diverse. The real revolution began with the development of manned submersibles, robust steel cocoons designed to carry human observers directly into the high-pressure environment. Early pioneers like William Beebe in his bathysphere in the 1930s and Auguste Piccard in his bathyscaphe Trieste, which famously descended to the deepest point of the Mariana Trench in 1960, offered humanity its first direct, albeit fleeting, glimpses of the abyssal world. Imagine the quiet awe of Bibi peering through a thick quartz viewport, describing the strange glowing forms that drifted past a living kaleidoscope in the perpetual night. These submersibles, though limited in maneuverability and dive time, provided invaluable direct observation, allowing scientists to witness deep-sea creatures in their natural habitat, observing behaviors and interactions that could never be gleaned from net-caught specimens. They were like fragile bubbles of human consciousness suspended in an alien ocean, collecting precious moments of direct, visual insight. Yet their very limitations, the narrow field of view, the brief dive durations, the inherent risks, underscored the immensity of the task, revealing far more darkness than light, far more unknown than known. The mid to late 20th century ushered in the era of remotely operated vehicles, or ROVs, which dramatically expanded our reach and endurance in the deep. Connected to a surface ship by a robust umbilical cable, which transmits power data and video, ROVs are essentially sophisticated underwater robots equipped with high-definition cameras, powerful lights, robotic manipulator arms, and an array of scientific sensors. Unlike manned submersibles, ROVs can remain submerged for days, even weeks, operating at extreme depths without risking human lives. They have become the tireless, unblinking eyes and dexterous hands of deep-sea science, meticulously surveying vast stretches of seafloor, collecting delicate samples, and capturing stunning high-resolution video footage of previously unseen organisms. The discovery of vibrant hydrothermal vent ecosystems teeming with unique life fueled by chemosynthesis was largely enabled by ROV technology, revealing entirely new paradigms of life on Earth. Each dive brings back a torrent of visual and environmental data, a digital tapestry woven from the threads of the deep. However, even with their advanced capabilities, ROVs are tethered to a surface vessel, limiting their range and making them vulnerable to currents and entanglement, leaving vast unexplored areas beyond their reach. Their eyes still illuminate only a small cone of darkness, like a flashlight beam in a colossal, unlit cathedral. Building upon the success of ROVs, autonomous underwater vehicles, or AUVs, represent a further leap in deep-sea exploration. These untethered robots operate independently, following pre-programmed missions across vast areas of the ocean floor, mapping topography with multi-beam sonar, measuring water chemistry, and even capturing images. Without the need for a surface ship's constant support, AUVs can cover far greater distances, exploring regions previously inaccessible due to their remoteness or the logistical challenges of tethered operations. They are the silent scouts of the abyss, systematically charting the unseen landscape and gathering environmental data that provides crucial context for understanding deep-sea life. An AUV like Sentry, for instance, can spend days mapping a hydrothermal vent field, creating detailed three-dimensional models of the seafloor and identifying potential sites for further investigation by ROVs. While AUVs are not typically equipped for complex sampling or close-up biological observation in the same way as ROVs, their ability to conduct broad-scale surveys expands our knowledge of the deep's physical characteristics, revealing new habitats where undiscovered life may thrive. Their quiet, methodical work reveals the hidden contours of the deep, hinting at the untold stories etched into the ocean floor. Deep-sea landers represent another crucial technological eye, offering a unique perspective on abyssal life. These uncrewed platforms are deployed directly onto the seafloor, where they can remain for months, even years, patiently collecting data. Equipped with time-lapse cameras, bait traps, environmental sensors, and acoustic recorders, landers provide invaluable insights into the long-term behaviors, foraging strategies, and population dynamics of deep-sea communities. They capture the slow, deliberate rhythm of life in the abyss, documenting creatures that might be too shy or too rare to be encountered by fleeting submersible or ROV dives. A lander might capture footage of a colossal amphipod slowly approaching a bait package or a deep-sea shark, investigating a whale fall over several days. These patient sentinels gather data that reveal the nuanced reality of life in the profound darkness, confirming the presence of creatures that would otherwise remain fleeting shadows. Their stationary presence allows for continuous observation, revealing the hidden patterns and long-term processes that shape deep-sea ecosystems. Beyond these primary platforms, a suite of specialized sensors and imaging systems further enhances our technological perception of the deep. High-resolution sonar systems mounted on ships or AUVs create incredibly detailed bathymetric maps, revealing previously unknown seamounts, canyons, and abyssal plains, each a potential habitat for undiscovered species. Advanced deep-sea cameras, often equipped with low-light capabilities, and specialized lighting arrays can capture stunning images and videos, piercing the perpetual darkness with carefully controlled illumination. Hydrophones sensitive, underwater microphones listen to the acoustic landscape of the deep, recording the clicks of sperm whales, the groans of unknown fish, and even the enigmatic ultra-low frequency sounds that occasionally hint at the presence of truly colossal undiscovered creatures. Environmental DNA or eDNA analysis. A revolutionary technique involves collecting water samples and then sequencing the trace amounts of DNA shed by organisms in their environment. This non-invasive method can detect the presence of species, even rare or elusive ones, without ever directly seeing or capturing them, offering a powerful new ear to listen for the genetic whispers of undisclosed life. Yet despite this breathtaking array of technological advancements, the paradox of discovery persists. The more we see, the more we realize the vastness of what remains unseen. Each new image, each new data point, each new species formally described opens up a cascade of new questions. We might discover a new species of anglerfish, but then we wonder about its full life cycle, its precise ecological role, its reproductive strategies, and its evolutionary relationship to other deep-sea fish questions that often require years of further painstaking research to even begin to answer. The deep ocean is not merely a collection of isolated organisms. It is a complex, interconnected system, and understanding one part often necessitates understanding many others. The sheer scale of the deep ocean continues to overwhelm even our most advanced technological eyes. We have mapped less than 1/5 of the global seafloor, with modern sonar and direct visual observation covers an infinitesimally small fraction of the ocean's immense volume. Imagine trying to understand the biodiversity of an entire continent by only observing a few square meters here and there. This analogy, though vast, still falls short of capturing the true scale of the deep ocean's unexplored territory. Furthermore, our technological eyes still operate within significant limitations. Artificial lights, while essential for imaging, can alter the behavior of light-sensitive deep-sea organisms, leading to observations that may not fully reflect their natural state. Robotic arms, no matter how dexterous, can struggle with the extreme fragility of many deep-sea creatures, making pristine collection a continuous challenge. Even the deluge of data generated by modern expeditions creates its own challenges. Gigabytes and terabytes of video footage, sonar maps and sensor readings pour in from every dive. Processing, analyzing, and interpreting this colossal influx of information requires immense computational power and dedicated human expertise. Identifying new species from vast video libraries, for instance, requires tireless, meticulous scrutiny by highly trained taxonomists, a resource that is already stretched thin. Many hours of valuable footage brimming with potential discoveries may sit, unanalyzed for years, their secrets still locked within the digital archives, waiting for the focused attention that might finally bring them into the light of formal announcement. As technology continues to evolve with the promise of more intelligent AUVs, lighter and more maneuverable ROVs, and even more sensitive sensors, our view into the deep will undoubtedly expand further. We will likely discover countless more species, perhaps even entirely new ecosystems or forms of life that defy our current biological understanding. But this expansion of knowledge will almost certainly be accompanied by an even greater realization of the ocean's vast, enduring mystery. The technological eyes we deploy into the deep are powerful, yet they are mere pinpricks of light in a colossal, three-dimensional darkness. They expand our view, allowing us to glimpse wonders previously unimaginable, but in doing so, they also reveal the profound, humbling extent of what remains hidden, deepening the mystery and reminding us that the deep ocean will continue to hold its most profound secrets for generations to come, patiently waiting for the patient, persistent gaze of humanity's ever-advancing technological eyes. Ethical dilemmas in deep-sea revelation. The pursuit of knowledge, particularly the unveiling of life's hidden diversity in the deep ocean, is often perceived as an unalloyed good, a pure quest for understanding. Yet with every new discovery, with every species brought from the abyssal darkness into the light of scientific awareness, comes a profound ethical weight. The act of revelation itself, of announcing a previously unknown deep-sea creature or ecosystem to the world, can trigger a cascade of complex moral and practical dilemmas, forcing scientists and policymakers to grapple with the potential consequences of their newfound knowledge. This is the delicate balance between the imperative to discover and the responsibility to protect attention that casts a long, thoughtful shadow over the very act of deep-sea revelation, sometimes leading to the deliberate, temporary withholding of information for the greater good. One of the most immediate and pressing ethical dilemmas arises when a newly discovered deep-sea species or ecosystem is found to be exceptionally vulnerable or particularly unique. Imagine a pristine hydrothermal vent field teeming with endemic species found nowhere else on Earth, a delicate oasis of life, powered by the Earth's internal heat. Or consider a newly described species of deep-sea coral, growing at an incredibly slow rate, perhaps only millimeters per year, its ancient structure representing centuries of growth. If the precise location and characteristics of such a discovery are immediately broadcast to the world, there is a legitimate concern that this knowledge could attract unwanted attention from various human activities. Research vessels, while conducting scientific studies, can inadvertently disturb delicate habitats. More significantly, the deep sea is increasingly viewed as a frontier for resource extraction, particularly for polymetallic nodules and seafloor massive sulfides rich in valuable minerals. The public announcement of a unique, thriving deep-sea ecosystem could inadvertently highlight a potential resource hotspot, drawing the attention of mining companies. In such cases, scientists might face the difficult decision to delay the formal publication of a discovery or to obscure its precise location in order to buy time, for the establishment of protective measures, such as marine protected areas, before irreversible damage can occur. The weight of this knowledge becomes a silent burden, a protective shield against potential exploitation. The collection of deep-sea specimens itself presents a series of ethical considerations. While essential for formal description and scientific study, the act of sampling inevitably involves removing individuals from their natural habitat. For species that are incredibly rare, slow-growing, or have extremely low population densities, the removal of even a single individual could have a disproportionately significant impact on the local population or even the species' long-term viability. Scientists must carefully weigh the scientific value of a specimen against the potential ecological cost of its collection. This often involves rigorous permitting processes, detailed justification for sampling, and a commitment to minimizing impact. The use of non-invasive techniques such as environmental DNA analysis or high-resolution imaging is increasingly favored when possible. But for formal taxonomic description, a physical type specimen remains a necessity, creating an inherent tension between discovery and preservation. The ethical imperative is to collect only what is absolutely necessary and to ensure that every collected specimen yields the maximum possible scientific insight, honoring its sacrifice. Furthermore, the advancement of deep-sea technology, while enabling unprecedented access, also amplifies these ethical concerns. As ROVs become more agile and AUVs more autonomous, our ability to penetrate even the most remote and pristine deep-sea environments grows. This expanded capability comes with an increased responsibility to ensure that our exploration does not inadvertently harm the very ecosystems we seek to understand. The lights of submersibles can disturb light-sensitive organisms. The noise of thrusters can disrupt delicate acoustic communication, and the physical presence of vehicles can alter the micro-habitat. Scientists are constantly striving to develop low-impact exploration techniques, but the challenge remains profound. The ethical dilemma here is not just about what we can do, but what we should do, and how to balance the insatiable human drive for knowledge with the profound duty to tread lightly on the last wild frontiers of our planet. The commercialization of deep-sea discoveries also introduces significant ethical complexities. The unique biochemical properties of deep-sea organisms, adapted to extreme conditions of pressure, temperature, and darkness, represent a vast, untapped potential for biotechnological applications. Enzymes that function at high temperatures, or pressures, novel antibiotics, or unique genetic pathways could hold immense value for pharmaceuticals, industrial processes, or material science. The discovery of a new deep-sea microbe with a unique metabolic pathway, for instance, could lead to a patentable product worth billions. This potential for bioprospecting raises critical questions about intellectual property rights, benefit sharing, and the potential for commercial exploitation of deep-sea genetic resources, particularly when discoveries are made in international waters beyond national jurisdictions. Who benefits from these discoveries should the wealth generated from deep-sea biodiversity be shared with all of humanity? Or should it accrue solely to the discovering entity? These are complex legal and ethical questions that remain largely unresolved, adding another layer of weight to the act of deep-sea revelation. The very act of naming a species, while a cornerstone of taxonomy, can also carry ethical implications, particularly when it comes to publicly accessible information. The choice of a species name, whether honoring an individual, a location, or a characteristic, can sometimes lead to unintended consequences. More broadly, the formal announcement of a new charismatic deep-sea species can generate intense public interest, sometimes leading to calls for conservation. before enough is known about the species' ecology or population status to implement effective measures. Conversely, the lack of formal description for a known but unannounced species means it cannot be included in conservation assessments, making it invisible to protective legislation. If a species remains scientifically undisclosed, it cannot be listed as endangered or vulnerable under international agreements, leaving it entirely unprotected, even if its existence is known to a small group of scientists. This creates a deeply frustrating ethical paradox. The very process of formal recognition, which is meant to protect, can be painstakingly slow, leaving vulnerable species in a perilous state of unacknowledged existence. Furthermore, the ethical responsibility extends to the communication of deep-sea science to the public. The deep ocean, with its bizarre and often frightening creatures, can easily be sensationalized. The narrative of sea monsters or alien life forms can fuel fear and misunderstanding, potentially hindering genuine conservation efforts, or leading to calls for invasive exploration driven by curiosity rather than scientific imperative. Scientists bear the ethical burden of communicating their discoveries accurately, responsibly, and in a way that fosters respect and wonder for these unique ecosystems, rather than fear or exploitation. This involves carefully differentiating between established facts and scientific hypotheses, acknowledging the limits of current knowledge and emphasizing the profound interconnectedness of deep-sea life with the health of the entire planet. Finally, there is the overarching ethical dilemma of humanity's impact on the deep sea. As our technological capabilities expand, so too does our footprint. Climate change driven by human activities is already impacting the deep ocean through ocean warming, acidification, and deoxygenation, threatening ecosystems that were once thought to be insulated from surface changes. Microplastic pollution has been found in the deepest trenches, and persistent organic pollutants accumulate in deep-sea food webs. Every discovery of a new species or ecosystem in the deep comes with the sobering realization that these pristine environments are no longer truly isolated, but are increasingly vulnerable to the cumulative effects of human activity. The weight of this knowledge compels scientists not just to discover, but to advocate for the protection of these fragile, irreplaceable realms, to ensure that the wonders they unveil are preserved for future generations, rather than lost to the unintended consequences of our own relentless curiosity and technological prowess. Thus, the journey of deep-sea revelation is not a simple path to enlightenment, but a complex ethical landscape fraught with difficult choices. The weight of knowledge is a heavy one, demanding careful consideration of potential impacts, a commitment to responsible stewardship, and a continuous re-evaluation of our role as explorers of the planet's last, true wilderness. It is a constant negotiation between the thrill of discovery and the profound responsibility to protect what we find, ensuring that the unveiling of the deep ocean's secrets ultimately serves to enrich rather than diminish the astonishing biodiversity of our dreaming planet. Dreams of the deep and discoveries yet to surface. As our understanding deepens, piercing through the layers of water and time, a profound realization settles within the quiet spaces of scientific contemplation. The deep ocean remains unequivocally the Earth's last true frontier, an endless expanse of unexplored territory and undiscovered life. Despite centuries of intrepid exploration and decades of advanced technological deployment, the vast majority of this colossal three-dimensional realm remains entirely unknown to humanity. We have in essence merely dipped our toes into the immense liquid darkness, catching fleeting glimpses of wonders that hint at a biological richness and geological complexity far beyond our current comprehension. The dreaming ocean continues to hold its secrets with serene tenacity, a boundless canvas upon which the future of discovery will undoubtedly unfold, perpetually inviting our curious gaze. Consider the sheer scale of what remains to be charted. Less than 1/5 of the global seafloor has been mapped with high-resolution sonar, leaving more than 80% of the ocean bottom veiled in topographical obscurity. This unmapped terrain is not merely flat, featureless plains. It encompasses colossal mountain ranges, deeper canyons than any on-land vast abyssal plains, and potentially countless hydrothermal vents and cold seeps, each a potential oasis for unique, undiscovered ecosystems. Imagine entire mountain ranges stretching for thousands of kilometers, lying hidden beneath kilometers of water, their peaks and valleys harboring species entirely unknown to science. The sheer volume of the water column itself extending from the surface to nearly 11,000 meters in the deepest trenches represents the largest contiguous habitat on Earth. A three-dimensional world where life exists from the surface to the sediment, yet remains largely unsampled and unobserved in its middle depths. This immense uncharted territory is the ultimate promise of the endless frontier, a silent invitation to future generations of explorers. The next wave of deep-sea exploration will undoubtedly be driven by even more sophisticated technological eyes, designed to overcome the persistent challenges of pressure darkness and scale. We are on the cusp of a new era of autonomous exploration, where swarms of intelligent autonomous underwater vehicles, or AUVs, will work in concert mapping vast areas of the seafloor with unprecedented detail and collecting environmental data with tireless efficiency. These untethered robots equipped with advanced artificial intelligence will be capable of independent decision-making, adapting their missions in real time based on unexpected discoveries, perhaps even identifying and tracking elusive deep-sea creatures without human intervention. Imagine a fleet of silent, glowing explorers systematically revealing the hidden contours of the abyss. Their collective data streams painting a comprehensive picture of the ocean's profound depths in a way that single, isolated expeditions never could. Furthermore, the development of bio-inspired robotics holds immense promise for more delicate and less intrusive exploration. Drawing inspiration from the soft-bodied, pressure-adapted creatures of the deep themselves, engineers are developing robots with soft, flexible exteriors capable of moving with unparalleled grace through the water column, minimizing disturbance to fragile organisms and sensitive habitats. These gentle machines could collect specimens with unprecedented care or simply observe life without altering its natural behavior, offering a truly unadulterated glimpse into the abyss. Coupled with advanced optical and acoustic sensors, these future explorers will provide clearer, more comprehensive data, allowing scientists to understand the subtle nuances of deep-sea life without the harsh intrusion of current methods. The potential for truly revolutionary biological discoveries remains immense. Scientists theorize that the deep ocean could still harbor entirely new phyla or even kingdoms of life, representing branches of the evolutionary tree that diverged from known lineages billions of years ago and have remained isolated in the stable, extreme conditions of the abyss. The discovery of life forms based on entirely different biochemical principles, perhaps utilizing novel energy sources or displaying unique cellular structures, is not beyond the realm of possibility. The extreme environments around hydrothermal vents and cold seeps, for instance, continue to yield organisms that challenge our understanding of life's fundamental limits, suggesting that other, even more exotic forms of chemosynthetic or extremophilic life could exist in yet-to-be-discovered geological settings. These findings could fundamentally alter our understanding of the origin and evolution of life on Earth, and even inform our search for extraterrestrial life on distant ocean-bearing worlds. The existence of truly colossal, undiscovered leviathans hinted at by the scars on sperm whales and the enigmatic acoustic signals from the deep remains a tantalizing possibility. The sheer vastness of the deep ocean combined with the phenomenon of deep-sea gigantism creates an environment uniquely suited for the evolution of immense, slow-growing and long-lived creatures that could easily evade our current sampling and observation technologies. Future exploration with its expanded visual and acoustic reach might finally unveil these hidden giants, bringing them from the realm of whispers and shadows into the undeniable light of scientific recognition. Imagine the profound impact of discovering a new species of colossal squid, dwarfing even Architeuthis, or an unknown lineage of deep-sea shark, its existence previously confined to the realm of speculative fiction. Beyond individual species, future discoveries will likely reveal entirely new types of ecosystems, driven by energy sources or biological interactions that we have yet to conceive. The deep ocean is a mosaic of habitats, each with its unique environmental parameters and biological communities. As our mapping capabilities improve, and as we explore previously inaccessible areas like sub-seafloor environments or the water column of the Hadal Trenches, we will undoubtedly encounter new biological hotbeds, new oases of life that operate under principles entirely foreign to our current understanding. These discoveries will not only expand our biological catalogue, but will also deepen our appreciation for the astonishing adaptability and resilience of life itself. The future of deep-sea exploration will also necessitate unprecedented levels of international collaboration and data sharing. The ocean by its very nature transcends national boundaries, and the challenges of exploring its depths are too immense for any single nation or institution to undertake alone. Collaborative efforts pooling resources, expertise, and technological capabilities will be essential for systematically exploring, understanding, and ultimately protecting this shared global heritage. The establishment of international deep-sea observatories, long-term monitoring stations equipped with a multitude of sensors and cameras, will provide continuous streams of data, allowing scientists to track changes in deep-sea ecosystems over decades, offering insights into the slow, deliberate rhythms of abyssal life that are impossible to capture through brief, episodic expeditions. Ultimately, the deep ocean represents not merely a physical frontier, but a profound intellectual and philosophical one. It challenges our anthropocentric view of life, forcing us to confront the limits of our knowledge and the astonishing diversity that exists beyond our immediate perception. Each new discovery, each species brought to light, each ecosystem unveiled serves as a humbling reminder of the immense, ongoing process of life on Earth, a process that began billions of years ago and continues to unfold in the quiet darkness of the deep. The untold stories, the unwritten chapters, and the unseen inhabitants of this dreaming ocean continue to beckon, promising an endless stream of wonders for those who dare to peer into its profound, silent embrace. For the deep ocean's secrets are far from exhausted, and its most extraordinary revelations are very likely yet to surface. And so, as we drift away from the profound depths of the dreaming ocean, let the lingering mystery of its unseen life gently settle within your mind. Remember that the very atoms composing those abyssal creatures, the strange, resilient forms we have contemplated, and indeed the elements that make up your own being, all share a common ancient heritage. The iron in your blood, the carbon in your cells, the oxygen you breathe each, was forged in the fiery hearts of distant stars, scattered across the cosmos, and gathered here on this watery world. You are in every fundamental particle, a child of the universe connected to the boundless unknown both above and below. As you surrender to the quietude of sleep, allow your own inner depths to mirror the ocean's vastness, a realm where thoughts drift like marine snow, and dreams unfurl like bioluminescent wonders. May the cosmic currents cradle your deepest dreams as the unseen ocean cradles its sleeping wonders. Sleep well. See you again.